Okay, my name's David Taggart. I'm from University of Adelaide. I became involved in this project, or became interested in this project, which is part of a much larger project looking at declining native grasslands in the Murraylands, because I'd worked at a site within the Murraylands up at Cooloola Station on wombats for the last 23 years, and had seen across that period of time a change in the vegetation up at the station we're working at. When we first arrived at Cooloola, there was an old farmer there who's now since retired and moved into another town in the Murraylands. But he was telling me when he first moved into Cooloola back in the 60s, he said in years of high rainfall, he said the native grasses were like a forest. And he said, a forest, these are his exact words, a forest the size of the height of my horse's head. And he said it was so thick and dense, the native grasses, that he'd lose his sheep in amongst the grass that he couldn't see them. And then later, when he got rid of his horse and brought his first ute, he said there were years that he could tie the grass in a knot over the bonnet of his ute. That's how long they were. So anyone that's familiar with the Murraylands, or from our generation, probably aren't familiar with seeing a site like that. So it was hearing those words and seeing what had happened across that 23 years that really got me interested in this project. And I'd also like to say that what I'm talking about today is very, very preliminary information, and the tame geneticist involved in this project is on holidays in WA, so don't ask me any genetic questions. Okay, so for those who don't know, Murraylands, situated west of the Murray River, south of Morgan, north of Madam, and east of the hills. Now within the Murray, it's of course found within the Murray-Darling Basin, has a high drought frequency up to 78% and low rainfall of 200 to 300 millimetres. There's been extensive clearing in the area of mallee scrub for grazing, timber harvesting, charcoal production, and that was primarily for the steamboats going up and down the river, and crop production. So the timber and charcoal activity stopped in the early 1900s, and the cropping now only occurs really close to the Mount Lofty ranges where the rainfall's more reliable. Grazing's traditionally been underpinned by the region's diverse native veg, particularly the grasses and forbs. So in this region, native vegetation is vital for the health and prosperity of the region's ecosystems and natural resource-based industries and communities. However, it's been impacted by a wide variety of threats, including clearing, loss of biodiversity, changed fire regimes, reduced water, salinity, exotic weeds, and overgrazing by diversity of native species and pest and domestic stock. Now, the aims of this study were to determine the seasonal effects, or seasonal changes in the diet and dietary overlap of the species grazing in these areas, to determine how the loss of native pasture affects the dietary preference of these species, and to determine the biochemical and nutritional value of the plants consumed and how this varies with season. Now, as I said, because it's such a preliminary study, a lot of this stuff I won't be covering today, but just to give you an idea of the breadth of the project when it's completed. Okay, the two study sites we're looking at is Cooloola, so that's the one we've been doing the wombat work on for 23 years, just near Swan Reach. So it's sheep grazing property, about 15,000 acres originally, although it's been fragmented now, broken up now. It's fragmented and degraded mallee, scrub, saltbush and bluebush understory, and the ground cover is primarily dominated by introduced weed species today, with some native grasses and forbs. And the veg at this site we consider average to poor in quality. Wonga Station, which is further north, just west of Morgan, again a sheep grazing property, a lot bigger, fragmented mallee, saltbush, bluebush understory, ground cover dominated by native grasses and forbs, very few weed species, and the native veg and herb layer is considered of quite high quality. So the common weeds at Cooloola, Ward's weed, thread iris, potato weed, onion weed, medic, horehound, and a variety of other weeds. In this system, or across both properties, you'll see red kangaroos, western grey kangaroos, Euros, southern hairy-nosed wombats, and introduced species, rabbits, sheep, and there's a Mediterranean snail that's present just on Cooloola, not up on Wonga, in their billions. So the methods we used, we established a plant reference library consisting of about 140 species. We did veg assessments from 15 randomly selected quadrats, each 20 metres by 20 metres on each of the stations. We collected scat and autopsy samples. So for the scats, we got 50 scat samples per species per season. The seasons we looked at were spring and late summer, and of course that was replicated on both stations. The autopsy samples involve collecting stomach contents and large intestine content from 5 animals per species per season. DNA is extracted from the plants using 50 milligrams of ground fresh leaf sample, and from the scat and autopsy samples, these are assumed to have a uniform content, and 20 milligrams of dried ground randomly selected sample was extracted or used to extract DNA from these. PCR amplification strategy followed that of Bell's, and using chloroplast barcoding primers to assess the DNA. A technique called MySequence sequencing and data analysis was used, which is, I'm told, next generation gene sequencing, and this work was carried out in the Croxford laboratory at the Plant Research Centre at the University of Adelaide. So some of the preliminary results from Cooloola, 142 samples were analysed by sequencing 2 chloroplast DNA markers. 7.5 million sequence reads were obtained of high quality using the MySequence information for plant, scat and autopsy samples. The majority of the Cooloola plant samples were able to determine them down to family, in most cases species. 9 plant species dominated the scats of all species examined, and it's mainly the scats I'll be talking about today. And preliminary analysis suggested that medic was the most commonly consumed plant species. This figure here is a figure showing the stomach and large intestine content from one individual red kangaroo. We haven't analysed the rest yet. The take-home message to note here is that there was the same plant species were identified in the stomach as were also found in a large intestine, which you may expect, but they're identified in differing proportions, and possibly due to differences in the digestibility and absorption of those different plant species. And now this hasn't been factored into the scat analysis at this stage, but will be. If you look at a summary of the percentage of the plant composition of the scats of the animals collected in spring at Cooloola Station, so it's a station with all the weeds present, and the yellow colour across the top just indicates some of the weed species, with the animal species down the side. What jumped out at us straight away is that if you're looking at the plant composition in the scats, the species that overlaps most closely with sheep were those Mediterranean snails, followed then by western grey kangaroos and wombats after that. It's also interesting to note that the diet of the wombats and the red kangaroos, or the plant composition of the scats, the wombats and the wombats and rabbits were quite similar, and also that the content of the scats of the red kangaroos overlapped least with sheep. If we look at summer, in summer we can see it's swapped around a little bit again. So in terms of their overlap with sheep, snails are still the species that overlaps most with sheep in terms of the plant content of their scats. Wombats, however, are now the one that overlaps least. And the wombats at this time are persisting mainly on wardsweed. And rabbits are the species that overlaps second most with that of sheep, and again have similar diets to wombats. If we look at the weeds, the proportion of weeds in the diet of the animals at Cooloola in spring versus summer, you can see that in spring Western grey kangaroos have the least amount of weeds in the scats at around 50%, but that goes up to 82% of their diet of red kangaroos contain weed species in spring. And if we then jump to summer, you can see that the animals are relying almost entirely on weeds to persist at Cooloola Station across the summer period. So just a summary of the preliminary findings at Cooloola. Although in all animals, all plant species are present in the stomach, were also present in the large intestine. Their proportions varied markedly. 9 plant species dominated the scats of all species studied across both seasons. Medic was the most commonly observed plant species in the scats across both seasons. The introduced weed species dominate the diet of all species examined at this site. The plant content of scats of the Mediterranean snails appears to overlap most closely with those of sheep, both in spring and summer. The plant content of red kangaroos scats in winter and southern herring-nosed wombat scats in summer show little overlap with that of sheep at those times of the year, and the plant content of rabbit and wombat scats overlap closely across seasons. Thanks very much.
Thanks, Dave. Time for probably one question.
So was the wombat suffering from eating these introduced species at all, or was the position of the animals very good?
Well, actually, that's interesting you should bring that up, Bill, because when we started in 1993 doing the wombat work, we'd go out and see in the order of 120 to 200 wombats a night. And then in the 2002 drought, the population crashed, and we're only seeing about 30% of the animals we'd see back in the early to late 90s. And that's despite the farmer destocking the property in 2006 and virus coming through in 1996. So you'd think with that less grazing pressure with no stock and rabbit numbers being right down, we used to be able to go out in 4 hours, fill a freezer full of rabbits, like 100 rabbits, 120 rabbits in 4 hours shooting. Now you'd be lucky to go out and shoot 10 or 20 rabbits for the entire night. So you've got a lot less grazing pressure, but a lot less wombats in the system. They often recovered, and we're thinking that's tied in with the declining native grasses and the overabundance of all the weed species. So.
All right, thanks, Dave.